Hi everyone, my name is Elizabeth Grande. I'm from the Human Biology Students Union, and I just wanted to notify you guys of an event that's happening this Thursday. So we're having our annual Ontario Medical School Symposium. So for those of you who are interested in med school or want to know what it takes to get in, we're having U of T's admissions officer, or the med school admissions officer, come to give a talk on like what you need, like GPA, extracurriculars, like prereqs in regards to courses. We're also having med students from U of T and McMaster coming to speak about what it's like to be in med school, as well as the Princeton Review. We'll be raffling a free MCAT course, so that's about $4,000 worth. So everyone who attends, you enter your name in the raffle and you get a chance at winning this free prep course. And then free dinner will be provided for all you attend. So it's happening this Thursday in MS 2170 from, I believe it's 6 to 8. So I highly advise anyone who's interested in med school to come by. I believe it was posted on Blackboard, and thank you guys.
Okay, I do highly recommend if you have the inclination of eventually going to med school, or you're not sure about whether or not you can go to med school, if you're worried about how competitive your GPA is, if you're looking to get a mentor, if you're looking to just find out some more information, it's a good opportunity for you to go and chat with individuals who are in med school or who are involved in the process. I wanted to talk to you a little bit about what we are currently limited by in science. So as I mentioned to you in the last lecture, there's a lot that we think we're really good at in science. There's a lot of stuff that we're incredibly good at in science. There's no doubt that a lot of the technological advances that we've had related to science have really improved the way that we do science. For example, a good one that I had when I was coming up through undergrad that is now taken for granted is something like PCR. All of you now do polymerase chain reactions. It's part of your undergrad course, and it was a technology that I had sort of seen being developed coming up through undergrad. And there's no doubt that now our knowledge of DNA, genomics, and everything that results from PCR is incredibly advanced as a result. But I'm going to go through two different cases with you, one related to placebos, and this goes back into things like complementary alternative medicine, as well as a limitation that we currently have. We'd like to think that right now science is at its apex. We are the best that we've ever been in terms of science, and as a result we should be able to do things like solve problems like Zika, the Zika virus, and understand it almost de facto. That we should be able to do that without even thinking or trying about it. But the reality is science right now has this major roadblock. There's this massive epidemic that is going on in the Caribbean, in South America and Latin America, that we simply have no understanding of. We try our best in terms of the science to understand it, but right now science is really limited. And that may be surprising to you, it may be shocking to you that science right now doesn't have all the answers that we think we do. We will go through some of those different issues in the next week or so, and maybe use science to fix this potential problem. So I'm going to go back in the first part of today's lecture to talk to you a little bit about placebos. The effects that we saw with different types of complementary and alternative forms of medicine actually do occur. Why do they occur? We have a scientific rationale for this. And in the second part, I'm going to talk to you about what science doesn't understand. We know that there is a link between Zika infections as well as different disorders like Guillain-Barré and microcephaly, but we don't know what the mechanism is. And that, again, as I've tried to point out several times, really bothers us as scientists. Like, we should know. It's like being a mechanic, seeing a car running, or hearing that a car doesn't run as well as it should, and not knowing what the problem is. As a mechanic, as a biologist, it really bothers us. So we're going to go through these two different scenarios, and I'm going to go back to what I talked to you at the end of Friday's lecture and talk to you a little bit about placebos. The placebo effect, the reasons why many of those alternative forms of medicine or the complementary forms of medicine, one of the reasons why we think that they work so incredibly well, even if there is no active ingredient, even though it says clearly on the label that this contains an extract from this particular plant, and it turns out after we look for it, there is no active ingredient at all, that it still works for many individuals because of this effect. And it is a very real effect. And this goes back into what I talked to you about in terms of placebo. Placebo, and this is the actual monograph, you can still find it. It is still cited in the Journal of the American Medical Association back in 1955 by Henry Beecher. Henry Beecher is one of those giants in the field of medicine. Henry Beecher is the person who came up with a standardized way to test if something is a placebo. He is also the scientist who came up with this idea that if you are doing biomedical research, you should actually take ownership and have ethics associated with biomedical research. Henry Beecher is a giant in the field of research, even though you may not be aware of all the things that he's done. And Henry Beecher was the first person to actually suggest that in an experimental setting where you had an active compound, a real drug in other words, and something that was an inert substance that shouldn't have any real effect or impact on your body, that when you did this in an experimental setting, that the inert substance, under certain conditions, actually had a physiological, biological response. And this was published in 1955, and it has withstood the test of time. We know that the placebo effect that he described in 1955, the powerful placebo, actually is a real effect. It is not a sham. It is not the animal magnetism that was being purported in the 18th century. This is a real effect. And follow-up studies using the same principle, that we have to do this experimentally, that we have to be able to control the conditions in which this is used, has really revolutionized over the last 20 years our understanding of how the mind works, how your brain works, how drug studies should be carried out, as well as understanding why some of the biology works the way that it does. I'm going to take a little bit of a detour before I get into the rest of today's lecture. I don't know if you guys like statistics, but actually in biology, statistics is a fundamental core. Our understanding of genomics, by the way, to say that this gene is associated with this disease, actually there are two things that you need to have. One is a good understanding of genetics. The other is a good understanding of statistics. And it's only with the advent of population-based statistics, and here's an example of population statistics. It is known as a Hedges G. There are many different types of biostatistical measures. Most of you will know things like a p-test or a student t-test. You might know some non-parametric statistics. And this is one example of a population statistic. You look at a whole bunch of people over a protracted period of time. And this was actually really surprising. And I'm not going to go through all of the details. This is some of the details from a bipolar disorder study. In this particular figure, it's exactly the same for depression. So bipolar, depression, etc. When you have those different types of conditions, one of the things that you do is you take medication. In most instances, you go in, you see your psychiatrist. Your psychiatrist prescribes a certain type of medication that you take it, and then you get better over time. But what Hedges G did was actually, it took a look at some of these different effects. It looked at, let's take a look at some of the best and most often prescribed antidepressant and anti-bipolar medications that are available. And let's take a look at the number of sites that were used, or you can look at this x-axis in terms of time. So this could be represented by 1980s, 1990s, 2000s, and 2010. And let's take a look at the overall effectiveness of these different drugs compared to the population in general. And so this is an antidepressant medication study. It's also used in bipolar disorder. And one of the things that you can clearly see is that there is a negative or an inverse relationship. So think a little bit about this. What this is really saying is that when you take a look at antidepressant medications and you look at the total usage, whether it's in the 1990s and you compare it on the right to the year 2010, what is the trend that you see? Where is it more effective? Where is the number higher? You guys are looking at the same graph as I am, right? Where is it higher? On the left or the right? It's much higher on the left. And what Hedges G actually suggests, and this has been replicated several times, that when we take a look at the overall effectiveness of antidepressant medication that is used to treat depression or used to treat bipolar disorder, as shown down here in the figure legend, that over time, or by increasing the amount of usage, that the antidepressant medications, things like Prozac and others, have actually decreased their effectiveness over time. So over the last 40 years, in other words, the effectiveness of Prozac in a population has actually decreased. Does that make any sense to you? It is a statistical measure of a population. Does that make any sense to you? Can you think of a reason why this might occur? And here's a little clue for you. Any ideas? Any guesses? Any wild attempts at what might be going on here? Because this bothered biologists for a long time. So if I'm suffering from depression in 2010, have I biologically changed? Am I different than the individuals that were given the same Prozac, the drug is exactly the same, back in the year 1980? No, I'm not any different. My physiology is roughly the same as the physiology back then. So what has really changed here? Any ideas? Yes. Okay, so do you know the mechanism by which Tylenol works on your body to relieve you of headaches? No? Okay, but it still works for you, right? Okay. So what else might be going on? So overall, the effectiveness is clearly going down. It's very, very clear. It's a negative or inverse relationship. Drugs now don't work in a population level as much as they did back here. Well, not to keep you in suspense too much longer, but when we're comparing this, and we just looked at this in the previous slide, what do we compare this to? When we compare an active drug, we compare it to a placebo. So what has really happened, which we don't understand, and your colleague is right, we need to understand these mechanisms. The placebo effect over time has actually been increasing. The drugs are identical. People are still the same people. But overall, our placebo effect in 2010 and 2016 is much, much higher. We have a higher rate of the placebo effect in our general population than we did back in the 1980s. Surprising. And so we need to understand the biology behind this Hedges-G population statistic. And surprisingly, this placebo effect is best described in a number of different types of situations. Does the placebo work? If I had a psychiatric disorder like depression, and I was given a sugar pill, an inert substance, by all definitions that we've come across, a placebo, if I'm given a placebo, I'm equally likely to respond to the placebo than I am to the antidepressant medication that has an active substance. If I have Parkinson's disease, if I am given a pill that says you're going to walk better after taking the sugar pill, in Parkinson's disease, according to Kirsch and Saperstein, and many, many others, including Benedetti, in Parkinson's disease as well, you are also going to be walking better if you're given a placebo, a sugar pill. There are many others, including things like analgesia. You feel better. You feel less pain if you are given a sugar pill than if you're given something like an opioid. Surprising? Maybe not. And we're going to look at some of the biology behind this. So Kirsch and Saperstein were the very first to come up with a potential mechanism for what is going on. And in fact, they were the first to report that in 1998, that the overall effectiveness that we see associated with antidepressant medications was just as good. You're just as likely to feel better if you're suffering from depression if you're given a placebo as opposed to being given an active drug like Prozac, etc., or sertraline or any other serotonin reuptake inhibitor. So it's almost as effective. Think about that for a second. How do drug companies test to see if their drug is actually working in experimental control condition, just like we saw with Henry Beecher? They always compare it to a placebo. The placebos in these particular disorders, Parkinson's, analgesia or feeling pain, and psychiatric disorders work just as well as the placebo. So we have to rethink how we look at different types of diseases and the treatment for these different types of diseases. So again, for whatever reasons, and we do not understand these, and it is something that biologists and psychologists really want to understand, the placebo effect in the general population, you are more likely as a group of individuals, for whatever reasons, to believe in the effect of a placebo than the generations that came before you. We don't understand that, by the way, and it's something that is of great interest to biology as well as in psychology. So what are some of the different factors that we know and can understand about factors that mediate placebo? A lot of it is really due to context. If you are given a pill by someone who you trust that will tell you that if you take this pill, and it's your doctor, you take this pill, you're going to feel better, you're more likely to feel better. There is a psychosocial context to what the placebo is actually about. So there may not be any active substances at all in that particular pill, but you'll feel better just because your doctor told you that you'll feel better. So think back to what we did in our own class. So we looked at fecal pills, for example, and supplementing with dried fecal matter. And you thought, for example, that I actually would be eating that. And so some of you were actually really grossed out by it. And a lot of that is the psychosocial context. If I was a better actor, some of you might even be throwing up. I don't know. But it does depend on this psychosocial context. And this is really some of the mechanisms that we think underlies this placebo, that there are some mechanisms, in fact, it's your brain that determines whether or not a placebo will work, that involves expectancy, that you're thinking that whatever my doctor is giving me is actually going to work to my benefit. That expectancy, activating certain circuits in your brain, will actually lead to you receiving a benefit. Some of that is also pure psychology. It's behavioral conditioning. The conditioning that you have this expectancy of feeling better when you go to the doctor, just by showing up, visiting, seeing the doctor, you're going to be physically feeling a little better than you were before you went to see him. And again, some of that is patient-physician interactions. So let me take you through how we know some of this. So this again is a controlled experiment, something that as biology students you should know, related to how we test for things like expectancy. How are you expecting this drug to actually work? How is this a psychosocial context? How do we know, for example, if there's an active substance or not? And this is what is often done. Some of you who have gone into the hospital and have been in the hospital overnight will have had this patient-controlled device, and in some instances, an automated pump delivery system. And one of the things that we know, and I showed you on that previous slide, when we talk about things like placebo, placebo works on lots of things, but placebo works best on things like Parkinson's disease, making you move better, mood disorders and psychiatric disorders, as well as pain or analgesia, the relief of pain. And so one of the things that you can do is if you are given the same drug, either by a machine, an automated machine, or you're given that same drug, the same dose, over the same period of time by a physician. In fact, one of the things that we know that happens if it is in this open setting where you have a physician giving this to you, an open application, versus you're not even sure when the drug is being given to you. It's just an analgesic drug, something to block pain, that's given whenever the machine decides to give it, and you're given exactly the same dose over the same amount of time, that you will always feel better after you see a doctor giving you the drug. And that has been shown time and time again. So if we look at different types of analgesic drugs, if we look at things like morphine, if we give it through a physician in an open setting, or if we do it under a hidden setting where a machine is giving you the same dose of the same drug over the same time course, you will feel better. And the pain reduction, you have a bigger pain reduction, you feel less pain if you are given morphine in an open versus a hidden automated delivery system. If we look at other types of opioids, like tramadol, for example, we see exactly the same effect. So we know things like analgesia depends on expectancy. You're expecting to feel better. You have the psychosocial context of your physician giving it to you that make you feel better. This is a placebo effect, okay? This is why we get the placebo effect. It shows us that expectation actually improves the analgesic effect. And this is all done through what is known as a visual analog scale. Have any of you gone into one of the research hospitals like Mount Sinai or to Toronto General or to Toronto Western, and they've asked you to take part in a study, and they've asked you, how much pain are you feeling right now? So on a scale of like 1 to 10, like this is the worst pain I've ever felt, sort of the pain of losing the NBA championship, poor LeBron James, or you're feeling really, really good because you're about to retire and you're going out at the top of your game like Kobe Bryant. So on that scale, how do you feel? And you're asked to indicate just by drawing a line where you're at, and then if you're given a drug in an in a hidden setting, that pain level might go down a little bit. Or if you're given it sort of in an open setting, it might go down even more. You'll feel better and better depending on your expectancy of your pain and the pain relief. And so this is sort of the way a visual analog scale works. This is what is being measured here. And one other thing that I'm going to point out in this particular slide. So all of the drugs that are shown here on the left are drugs that look like opioids. We know that in our body, we have an endogenous system, the opioid system, that works in our brains to control neurotransmitter release, etc. And so does the same thing work? Does pain analgesia work when we're not looking at opioids and we're looking at synthetic drugs, non-opioid type drugs like ketorolac? And yes, it does. So this is also used in pain relief. And ketorolac, which is a non-opioid analgesic, also works in exactly the same way. So there is expectancy of pain relief. The doctor makes you feel better, and that expectancy definitely pays dividends in terms of the placebo effect as well. But how does all of this work? Pain and many other features that I've just shown to you, psychiatric disorders, things like Parkinson's, as well as analgesia, rely really on two different systems. I'm not going to go into this system very much. This is a reward-based system. So you feel a reward if this system, the dopaminergic system on the left, is activated. And there certainly is some reward-based expectancy. So if this system is activated, for example, you will feel better. If your VTA, the ventral tegmental area in your midbrain, becomes activated and it releases dopamine onto the nucleus accumbens, or NAC, then you will feel better. You will feel less likely to feel pain. You will have an expectancy next time that you go to visit your doctor that you'll be feeling better. So we know that the dopaminergic system plays a role in expectancy as well as in pain. But the true effects, the really big effects, especially in pain medication and analgesia, really depend on this descending pain pathway. Just again, out of curiosity, depending on how many of you have had this already, how many of you have taken PSL 300 or one of the second year PSLs? So many of you, about half of you. We feel pain in two different ways. There has to be a signal from the periphery that goes up into your brain, and that's an ascending pathway. And we also have a pathway that comes down from the brain to try to control pain. And pain always is integrated or put together at the level of the spinal cord. So imagine you've just hurt yourself or you're feeling pain. The only way that that pain signal can get up to the brain is through the spinal cord. It's the only way that nerves can really traverse. So think about this descending pathway. Think about how this opioid-ergic pathway is coming into play. So in the dorsolateral prefrontal cortex, so if you're looking at your eyebrow ridges, this is the orbital frontal cortex, and just to the sides, near your temples, you have the dorsolateral prefrontal cortices. Now this is the beginning of the descending control of pain pathway. And it sends one projection over into the hypothalamus, and we're not going to worry about that particular pathway. And then it sends another group of axons, and this is what these little bars represent. They send other groups of axons down into a region known as the periaqueductal gray. From there, other neurons will then, in the periaqueductal gray, will then send their axons to the rostral ventral medulla. And in your brainstem, at the level of the medulla, they will then send projections down into the spinal cord. And in theory, if you're feeling pain and you want that pain to stop, this is the pathway that you are activating. You are turning on the dorsolateral prefrontal cortex, which then in turn turns on things like the periaqueductal gray, which in turn turns on the rostral ventral medulla, which then stops signals at the level of the spinal cord. And that's the way that we think pain analgesia stops. And we know that the opioid system, as a neuromodulator, works at these different levels. So when you have an opioid drug, it acts to enhance this descending pathway. And this can inhibit those different signals by activating or making sure that this descending pain signaling pathway becomes activated. Now, I just told you that placebos An inactive substance, not an opioid, sugar, can activate the same pathway. How might it do that? How can an inert substance that doesn't contain opioids, which would normally stimulate this pathway, and stimulating it makes a lot of sense, how does a placebo actually drive this pathway? We're not 100% sure, but we know that we can actually block the effects of placebos if we give drugs that will block opioid receptors. So we know that opioids are released at these different synapses, and we know that if you are feeling good, you've just been given a sugar pill, your pain levels are going down, you're feeling much, much better after being given a sugar pill, and then we give you a shot of naltrexone or naloxone, which blocks opioid receptors, your pain levels start to go back up. Your placebo effect, there's no active substance, the is now being wiped out. And it's because it's blocking these opioid-ergic pathways. There's one other group of neurons that I'm going to point out to you here. And this group of neurons is really important, not just at this level, but also at the dopaminergic level. We know that neurons that release cholecystokinin, or CCK, and that is a type of neurotransmitter. So CCK, cholecystokinin, is also involved in this placebo pathway. If we activate neurons that contain cholecystokinin and it releases cholecystokinin, it's working just like naloxone as well as naltrexone, and it can block the effects of the placebo. So if you're feeling good after a sugar pill and you don't have naltrexone or naloxone around, you can certainly activate CCK groups of neurons, which will also block this placebo effect. To make things more complicated, if I were to take this group of students in this class, and let's say we have roughly 200 students in this class, about half of you would actually be really, really good placebo responders. If I gave you a sugar pill and I told you it was going to help you feel better after you stub your toe, half of you would respond well. Okay, yeah, you know what? I feel better after I ate that pill. The other half would not. And we know that there are some genetics that are involved in this system as well. And we know that when we start looking, that individuals that have certain single nucleotide polymorphisms, or SNPs, are better responders than others. Interestingly, or perhaps not interestingly, or surprisingly, or perhaps not surprisingly, a lot of this goes back to different neurotransmitters like serotonin, as well as some others. So if you have certain genetic variants of serotonin and you carry one of those single nucleotide polymorphisms, you may be a better placebo responder than the neighbor sitting right beside you. One of this is related to the synthetic enzyme that is involved in creating serotonin. So there are two different variants of the tryptophan hydroxylase 2 gene. And the promoter region has a polymorphism at position 703, and you either have a glycine or you have a threonine variant of this. And if you are homozygous for the G703 variant, you are going to respond better. If you get genotyped and it turns out you have G at position 703, you have a G residue, you are better at responding to placebos than if you have a T at that particular residue. It is also true, not just of the gene promoter at this one particular 703 position, but if you have the long version of the 5-HTP LR promoter, then you are also a better placebo responder. So if you get genotyped, and it turns out that your genotype comes back, that you have a long variant of this promoter and you have the G variant of the serotonin gene promoter in the gene promoter region, you are a much better placebo responder than someone who does not have either of these variations. In addition to that, we know that there are other genetic markers that are also in play. Individuals that have higher MAO, or monoamine oxidase A activity, act differently than individuals that have lower activity. So if you have high activity of this particular enzyme that breaks down neurotransmitters, you have a lower placebo response, and you likely have the GG polymorphism. So you can either have a G, a GT, or a GG, GT, or a TT within MAO. And if it turns out that you have a GG double polymorphism, then you are going to be having a lower placebo response. This goes further to other neurotransmitters. One of these is the catechol-O-methyltransferase, or COMT. If you have different alleles that are associated with this, at position 158, you will have lower overall enzyme activity, and if you have the version, you have the lowest overall activity, and if you have low activity of this enzyme, you actually respond the best to placebos. So this has actually caused the drug industry a lot of issues right now. It's costing the drug company lots and lots of money. If I know, for example, or I want to test the effectiveness of a drug, and I need to compare it because we talked about this in the last lecture. In order to be able to tell you if this drug is really effective or not, we have to be good biologists. In the past, what we've had to do is say that compared to an inactive substance like a placebo, that this drug works a lot better. I've also told you that for whatever reasons, placebo responses are going up. So drug companies now, before they start a drug trial, have to be able to eliminate all these super responders for placebos. So if I know that you're going to be a really high placebo responder and are going to mess up my results, I need to genotype you. I need to see what your COMT levels are like. I need to know what your serotonin transporter levels are like. And I need to exclude you from my study because I know that you will mess up my overall drug effect versus the placebo. And again, surprisingly or not surprisingly, these are the same types of markers that are associated with psychiatric disorders. We know, for example, that if you have social anxiety, that you have many of the same sorts of genetic variants. If you are suffering from depression, you're more likely to have some of these genetic variants. And so it turns out that there probably is a good biological influence for both. Just out of, again, curiosity, how many of you have had some sort of neurobiology course? Not too many, okay, a couple of you. Okay, I won't go into a lot of detail. So what I need you to make sure that you are aware of in this particular slide is that our brains, which consist of these structures here, these neurons, actually have to synthesize neurotransmitters. We don't make these usually. We can make do with some things like glutamate, for example, but others we need to be able to synthesize. Your brain doesn't normally have a lot of this neurotransmitter called 5-HT, which is serotonin. So we have transporters on our neurons that will transport basic amino acids that can get up into the blood-brain barrier, past it, and into the brain, and it gets into the neuron, and this tryptophan is then converted by that same enzyme we looked at before, THP2, into the first precursor 5-HTP. It then undergoes aromatic decarboxylation. You don't have to worry about all of that, into the final neurotransmitter, which is known as serotonin or 5-HT. And this is why we think these systems are involved in things like placebos. These are the same neurotransmitter systems that are involved in social anxiety, depression, as well as a number of other psychiatric conditions. And we won't worry about the other details on this slide about the different variants of the 5-HT postsynaptic and presynaptic neurotransmitter receptors. More or less, just to give you an overview that this is why we talk about THP2 and why we have to worry about neurotransmitter systems. We know that these systems, by the way, are involved. We know we can block them with certain drugs, naltrexone, naloxone, activating CCK pathways. We know that, for example, this involves these specific groups of neurons. So one of the ways that we know this is that in a neurodegenerative condition known as Alzheimer's disease, one of the first areas of the brain to go is the dorsolateral prefrontal cortex. The temporal lobes are also affected, but this is one area that's affected in Alzheimer's disease. So in Alzheimer's disease, we have this really interesting phenomenon. So this area of the brain starts to undergo degeneration. We lose this group of neurons, which begins that descending opioid-dependent pathway. So we can take these Alzheimer's disease patients, and one of the things that we can do is, if it's 2016 today, and we take a person or an individual who we know has Alzheimer's disease but is very, very functional, and we give them what is known as a frontal assessment battery test. We look for cognition, like do you recognize what this picture looks like, etc. And we give them a placebo. They respond really well to that placebo today. Now, 1 year from now, in 2017, if that same individual has come back and that same individual has had a degeneration in the dorsolateral prefrontal cortex, which all Alzheimer's disease patients will have after a year, and we give them the same frontal assessment battery test that they took today, we will see that their scores go down. And when their scores start going down and their dorsolateral prefrontal cortex is being measured by this score, the other thing that we'll note is the placebo doesn't work anymore. So this is one of the proofs that we have that the dorsolateral prefrontal cortex is really important in the beginning of this opioid-dependent descending placebo pathway. Again, we've already talked about naloxone and naltrexone as part of the opioid pathway. They are anti-opioid receptor blocking drugs. And we know also that we can do this in a non-pharmacological way. If I wanted to know if this pathway was really involved, another way that we can do this without giving you a drug or doing anything else is to use a technique in neurobiology known as repetitive transcranial magnetic stimulation, or TMS. And what we can do is we can put a big magnet over an area of your brain, like the dorsolateral prefrontal cortex, and stimulate that area of the brain, or turn off that area of the brain, depending on how we do this. And so when you are given a drug that is a placebo, or a placebo in place of a drug, and it produces analgesia and your pain tolerance goes up, we can block that by putting a large magnet over parts of the dorsolateral prefrontal cortex, again, as another form of proof that this is actually really important in the beginning of this pathway. One of the reasons why we use magnets is that the skull is actually not so great at conducting electricity. If you put an electric current or direct current, actually you have to ramp up the current quite a bit in order to get good effects on neurons inside your skull. A magnet does not have that issue. Have you ever done Faraday's rule in high school physics? Or have you ever taken a magnet and put it up near a TV and seen the changes in color that it produces on that TV? It's actually the same thing when you take a large, very powerful magnet and put it over different areas of the brain. Your brain works on electrical impulses. And we know through Faraday's rule that if you have a magnet, it can actually induce an electrical current in whatever wire you are putting that magnet over. In this case, the wire is your brain. And one of the things that we know is that this Faraday's principle can actually impact, turn on as well as turn off different areas of the brain. So what I just presented to you was how we use TMS, transcranial magnetic stimulation, to turn off different areas. We can also turn on areas. We won't go into that today, but these magnetic fields work very powerfully and very deeply in your brain. Right now, we're limited. We can't go any further than about 3.5 centimeters. After that, the overall effect of the magnet in producing a change in the electric field goes down precipitously. But structures that are really close to your skull, like your dorsolateral prefrontal cortex, we can put large magnets there and produce quite a substantial change on the left of the electrical fields that are being produced. So right now we're limited by the effect that TMS has to the first few centimeters on the surface of your brain. We're not entirely sure because it's a fairly new technique. We know that it can be used in research. It can be used in rehabilitation. So individuals like Jonathan Downer over at Toronto Western Research Institute use this to treat things like depression, anxiety, etc. But we don't know its long-term effects. We really haven't been able to study it for long periods of time. And we don't know if we increase the magnetic field, there's always a hint that there might be some issues with it. Okay, so I'm going to change gears now, and over the last 5 to 10 minutes or so, talk to you about some of our limitations. So science was great. It helped to understand the placebo. People were working on it since the 1950s to see why is this phenomenon actually occurring. So we don't know everything about it, but we've made a lot of progress. We know that there are certain single nucleotide polymorphisms. We know that there are structures in the brain that are involved. We know that we can block this effect using different types of drugs. But right now, in terms of science, this is one of the biggest mysteries that we have. We know that something is going on. We know that this is moving northward, and many of you have heard of Zika. And Zika, in many ways, is sort of a frightening proposition that science, right now, the best science that we have, can't answer all of these ongoing biological mysteries. So some of you may know, I'm originally from Brazil, and as a Brazilian, of course, you have to be cheering for your home side, and so I was like expecting 2014 when the World Cup was in Brazil, I was be out there celebrating that we won the World Cup, and unfortunately, didn't turn out that way. But at the same time that this was going on in Brazil last year, a few years ago, in 2014, at the same time, someone, somewhere, from some country, came into Brazil who was infected with Zika. They probably didn't know that they were infected with Zika. They probably didn't know or have any of the symptoms of Zika. But at some point in the World Cup, someone came into Brazil that was infected with the Zika virus. They may have been feeling okay. They may have been feeling really weird that if they didn't go, that they would be losing out on these $1,000 seats. For whatever reasons, an individual who's infected with Zika eventually made their way into Brazil and started infecting mosquitoes who were drinking their blood with Zika virus, which then spread. We had to wait about 9 months to see the overall effect of what Zika infection actually looks like. So we had no idea that this was going on, sort of under the table, behind the scenes, that these infections were going on until it was too late, until the very first individuals that had delivered their babies actually started to have some issues associated with it. And sorry, if there are any Germans here, I won't make fun of the German side. Great side. Congratulations on 2014. We'll see you in 2018. Maybe we'll have a little bit better luck. So let me tell you a little bit about Zika. So we know a lot about its overall pathogenesis. We know that in 1947, it was the first in Uganda. They will they feed to an uninfected individual. And we know that in particular that some mosquitoes are better carriers than others, and particular members of the Aedes family that I'll get into some more detail in the next lecture as well. And again, the overall transmission of the virus was relatively slow to begin with. 1947 in Uganda, in a isolated forest. We know that it started to spread through Micronesia. 2007 was the next big outbreak. And then 2013, 2014 in a very small population. And we know that somehow, in some ways, these two outbreaks in Chile as well as in Brazil are also somewhat related as well. And we need to think about this a little bit because it gives us a lot of pause. Like, why didn't we really care, for example, when individuals that were reporting Guillain-Barré syndrome, a very serious disorder, by the way, why did we not care when it started to make its way and become really predominant in Micronesia and parts of Micronesia? Why did it take this outbreak in Brazil before we started to really take notice? So after the World Cup, in April of 2015, this was the beginning, the first signs that there was something going on in Brazil. Typical symptoms, not unlike dengue, which is a normal endemic part of being in Brazil. You get bitten by a mosquito, you may or may not have things like dengue. And so they're very, very similar types of symptoms. So you'll develop a rash, you'll have some pain at the site that you were bitten, you might have a maculopapular rash, large swelling, and one of the most telling signs that tends to distinguish it just a little bit from normal dengue is you might have conjunctivitis. Have any of you had conjunctivitis? What part of your body is affected in conjunctivitis? So it's your eye, your conjunctiva, the outer part. If you've ever kind of seen someone with like red eye or that's sort of the conjunctivitis that we're talking about is often associated with both dengue as well as with Zika. The problematic thing is, for whatever reasons, in terms of epidemiological studies in Brazil, and going backwards into Polynesia and parts of Micronesia, there is an association, we don't know the cause, there is an association between being infected with Zika at a certain time as well as having microcephaly in any babies that are born. A smaller head, a smaller brain. More likely that these individuals for the rest of their lives will suffer from seizures, developmental delays, etc. What a horrific thing to have when you're told that you are pregnant in Brazil right now and maybe you might have been infected with Zika, and for the rest of your life you're going to have to be caring for a developmentally delayed child. Again, another interesting thing is we think that Zika is involved, but we have much better evidence that Zika is involved in Guillain-Barré. So yes, maybe there's a link with microcephaly and Zika infections, but we have stronger evidence to suggest Zika infections lead to a syndrome known as Guillain-Barré syndrome. Both, by the way, just again to point this out to you, is something that affects the nervous system. So microcephaly is where you have a smaller head, and I'll give you some examples on Friday. Usually, when we see examples of microcephaly here in North America, instances of microcephaly where we have an underdeveloped brain, an underdeveloped cranium, a smaller head overall, is really due to chromosomal abnormalities. It might occur in rare instances where we have decreased oxygen to the fetal brain, cerebral anoxia, that might be occurring at different time points. We know, and this is very surprising, we know, for example, that if you have infections of the fetus during pregnancy, that other types of viruses and bacterial and other zoonotic infections were also implicated in microcephaly, things like chickenpox or varicella, rubella, or German measles, toxoplasmosis. Cytomegalovirus was the first instance where if you had a cytomegalovirus infection during pregnancy, that the newborn would have microcephaly. So there was a viral link. No one had ever reported Zika as being involved in this. And these are not, by the way, those positive-stranded, single-stranded RNAs. These are very different types of viruses. We know that other things, exposure to drugs, malnutrition, fetal ketonuria, can all cause microcephaly. None of the women in Brazil had tested for these. The only thing that they tested for was Zika. Again, really surprising. It sort of was at the limits of what we knew in terms of biology. So the last thing that I'll sort of leave you with before I get into what I'm going to cover on Friday. We know that Guillain-Barré is normally an autoimmune disorder. And what happens in Guillain-Barré is that if you ever have had those pins and needles feeling in your legs, if you've been sitting down for a while or you've been kneeling for a while, imagine that you have that all the time. And this is what Guillain-Barré is. And it can start to spread. It can spread from one limb to another. It can start spreading upwards. It can affect your ability to breathe. And in some instances, it may actually result in paralysis. There are many different forms of Guillain-Barré. So there are forms that are due to acute inflammatory demyelinating forms. There is another form that I'll get into in some more detail called Miller-Fisher syndrome. There are ones that involve axonal neuropathy. There are ones that involve the sensory system and motor neuron systems more than others. But overall, it's something that affects your nervous system. And again, anecdotally, and this is in the literature, anecdotally, some individuals reported that Guillain-Barré syndrome, where you have these pins and needles, then it starts spreading across your limb, and from one limb maybe across a whole quadrant of your body. In some individuals, they have had some other prior infection before they started exhibiting these types of symptoms. Whether it's a respiratory infection or the stomach flu, something went on. And one of the possible mechanisms of this is that the Zika virus shown here may actually infect certain types of neuronal progenitor cells that we'll take a look at, or these neural stem cells. And it can either cause them to die, that's one possibility, or it may cause them to not develop appropriately. And we're going to look at both of these theories in this recent paper that tried to explain what happens when the Zika virus actually infects human cortical neural progenitor cells. And is that the reason why individuals in Brazil have babies with really small, underdeveloped cranium as well as the brain? And we'll talk about this on Friday.